We've got three types of viral infections we're going to talk about: persistent, latent, and oncogenic. Okay? So, persistent infections. The cell will harbor the virus and it's not immediately lysed. So, that exploding cell thing doesn't happen right away. Basically, it can hide out in the cytoplasm, or we can take that DNA and insert it into the host's DNA. So it's a way of hiding it. If it's part of the host DNA, it's not like there's anything weird hanging around. This can last from a few weeks to a lifetime. Uh, they may take a longer period of time, even years, for symptoms to show up, but the viruses are detectable in samples. So, two examples: HIV and HPV. So, human immunodeficiency virus and human papillomavirus. So, HIV, one of the things that made it really hard to identify at the beginning was that it would go in and it would release its DNA into the cell, and that would go and insert itself into the host's genome. So there was never any indication that there was a virus around because it was hiding. The other thing that made it kind of a challenge was you would get it, it would sit there, and it could sit there for a year, it could sit there for 6 months, it could immediately start making virus. There wasn't necessarily a perfect timeline of how it was going to work. So with HIV, once you have it forever, right? So it basically, time wise, how long it takes for symptoms to show up, it varies because it has that ability to take its genetic material, insert it into your genetic material, and just hide out. Okay, latent infections. Viruses go into a period of inactivity and can be incorporated as provirus. Then they emerge when you undergo stress, when you are in poor health. During that time of inactivity, when they kind of go dormant, there are no symptoms, there's no detectable virus in your body. It can cause recurrent disease. So I don't have any symptoms, I get super stressed out, it pops up again. Then, when I de stress and have some time to rest, symptoms go away. Okay, no symptoms, and then I get stressed out again and it pops up again. The most, I guess, well known of these is herpes simplex virus. So, you know the cold sores you get around your mouth? That's herpes simplex virus. So, it's in about 80% of the population. It's not nothing that you're like, oh my God, you're 80% of the population has this. But you'll notice that you get a fever blister, usually when you're stressed out. Finals ends, you get to have like a week break and whatever goes away and you're fine. And then the next semester starts and you get another cold sore. It is something that when it's not present on your face, you don't have viral load, you don't have any symptoms, there are no fever blisters, nothing. No one would know any better, but it pops up again. So it's latent for a period of time and then it comes back. And then it's latent for a period of time and it comes back. Herpes is kind of the best example, the most, I guess, common and well-known example of something that you still have. You still carry herpes virus, but it goes away and comes back. All right, oncogenic effects. Oncogens are usually cancer-causing agents. That's what that term means. So these are viral infections that can cause mutations in the host cell's DNA that result in cancers. Okay? So one of the most well-known right now is going to be human papillomavirus, HPV. Human papillomavirus is known for causing cervical cancer. It invades the cells of the cervix. It can cause mutations and cause those cells to become cancerous. Another example would be hepatitis B. Remember, hepatitis infects the liver. So it goes in, it causes mutations in the cells, and you've got tumors growing in your liver. HPV, it goes into the cells, it causes mutations, you've got cervical cancer. So you kind of have basically the onset of cancer because of the viral infection. All right, so bacteriophages. Bacteriophage kind of looks like a spider, okay? But as the name sounds like, okay, this is a virus that infects bacterial cells, okay? It can confer pathogenic properties to the bacteria they infect. Literally, it can pick up one trait from one place, and when it injects into another cell, it can take that trait with it. It has a similar life cycle as animal viruses, but it is a bit simpler in the fact that this whole thing doesn't get taken into the cell. Instead, it's almost like a hypodermic needle, okay? The legs and this spring here, when this is going to look weird, but when the spider sits, okay, it literally injects the genetic material into the bacteria that it is infecting, okay? So I don't have to uncoat it. I don't have to engulf it and then, or endocytose it and then release it. Literally, this genetic material just goes, okay? There are two types of bacteriophage life cycles, lytic and lysogenic. So lytic, remember, lysis means to break. So lytic, it's similar to the animal virus life cycle, and the release of the virus occurs through lysis, okay? Bursting of the cell. This thing, or this type of bacteriophage, kills the cell because I fill it up until I overinflate it and it pops and bacteriophage go everywhere. The lysogenic cycle is a bit different. They enter the bacterium, and kind of like HIV, the genetic material inserts into the bacterial chromosome. They form something called a prophage, okay? Sometimes you'll get induction to occur where it activates, okay? The prophage activates, it begins to replicate, and basically shuffles over to that lytic cycle. Start making copies until the cell explodes. But if it goes into that lysogenic cycle, it's less damaging to the host cell, okay? At least until it activates and begins replication. Lysogenic viruses can produce a phenomenon in bacteria, in the bacteria that they infect, called lysogenic conversion. Okay, prophages can pass along traits that let the bacteria produce a toxin or an enzyme that can help them with progression of disease. So, for example, diphtheria toxin, which is responsible for the seriousness of the disease diphtheria, is passed to the pathogen Corynebacterium diphtheriae by a lysogenic bacteriophage. So remember, when I insert my genome into another genome, when I cut it out to make it back into my bacteriophage, I cannot cut accurately. So I may take a piece of that genome from the bacteria, package it up into my bacteriophage, and insert it into the next cell. Okay? The cutting of that genome doesn't necessarily or isn't necessarily always I cut my perfect sequence out. Sometimes the cutting is off. Okay. So, another quick video.
The life cycle of bacteriophage T2 begins with a bacteriophage particle binding to the surface of the bacterial cell. The phage particle injects its genetic material, or DNA, carried in the capsid of the bacteriophage into the host cell. Once inside the cytoplasm, genes in the phage DNA direct the degradation of the host cell DNA and are able to utilize proteins within the host cell for the synthesis of new T2 phages. First, many copies of the phage DNA are made. The phage DNA encodes the proteins which form the capsid, and the regulatory proteins which direct their production and assembly into phage coats. The newly made capsid proteins and phage DNA molecules assemble into a new generation of phage particles, and the cell is lysed, releasing the mature phage particles.
Okay, so again, if we go back, that would be the lytic cycle. I inject, I start assembling, I make enough until the cell explodes. Okay? Okay, this one's different. This one is the lysogenic cycle. Okay, the one that it kind of inserts itself and sits dormant for a bit. Excuse me. Okay.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles.
Now, lysogenic.
In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
Okay. So you can see where it gets injected, immediately starts making the virus lysis. It gets injected, it works itself into the host's DNA. That host may, like it said, replicate over and over and over again, and every copy is going to have a copy of that phage DNA. So, other non-cellular infectious agents, prions. Prions are a misfolded protein. There is literally no genetic material in it. So they don't have DNA, they don't have RNA. It is a protein exclusively that is not folded correctly. How they replicate is kind of being investigated. What we do kind of know is that if you get a prion, the misfolded protein, it goes into your body, and for some reason, it induces your properly folded prion protein to misfold. So you've got this incorrectly folded protein causing your correctly folded proteins to all of a sudden misshape. Okay? It's associated with causing diseases known as spongiform encephalitis, okay, or encephalopathies. Uh, these diseases are progressive and fatal, and all of them affect the brain. Most human versions are genetically linked. So, Creutzfeldt-Jakob disease is a well-known prion disease that is genetically passed. Okay, so you get to a certain age, and all of a sudden, these proteins start misfolding. Brains of people who have prion disease look very similar to those that have Alzheimer's. The brain literally starts to lose mass, and parts of the brain start to kind of disintegrate. Okay. A variant of Creutzfeldt-Jakob is a human spongiform encephalopathy linked to bovine spongiform encephalopathy in cattle. So I don't know how young you are, but I want to say it was in the 90s. There was an outbreak of prion disease in Europe. So remember that Creutzfeldt-Jakob is really specific, it's genetic, and all of a sudden, all of these people were showing up and being diagnosed as having Creutzfeldt-Jakob, whose family history never showed any Creutzfeldt-Jakob at all. So what was happening? Um, After investigation, they found out that ranchers were actually feeding their cows the parts that weren't being used at the butcher of the cows to increase their protein intake, to increase their muscle mass. Cows are not carnivores. That's one. The other problem was that at some point, one of the cows that they butchered and put into the feed for the cattle had um, bovine encephalopathy. So when they fed that cow's brain, spinal cord, to the other cows, they basically gave them the misfolded prion. So they would start showing symptoms. Ranchers don't want to lose money. So what do? Take that cow and have it butchered. Give me the extra pieces so I can feed it to my other cattle. And you're passing it on again. So in the 90s, when this became a thing, they called it mad cow. They called it mad cow disease. And the, I guess, international shipping of European cattle, they were like, nope, we will not take any shipments of cattle from Europe anymore in the United States, because they literally couldn't tell if one of the cows had prion disease, and they didn't want it to come to the United States. So during that time frame, you're talking about 20-year-olds who are exhibiting Alzheimer's symptoms. Doesn't make any sense. Now, this is the bad part about prions. I'm going to sit down for this. So you know how with viruses and with bacteria, we can scrub down with bleach, we can use Lysol wipes, we can use hand sanitizer. Prions don't work that way. You can set a desk on fire and it'll kill bacteria and viruses. Prions will survive that. Straight bleach, hydrogen peroxide, prions will survive that. Remember, they're not cells. It's a protein that is misfolded. It is scary how well prions survive. So these ranchers that had these mad cows lost everything. The government finally stepped in, took whole herds of cattle, and slaughtered them all. And if I'm not mistaken, buried them. So, yeah. You can destroy prions, but you have to have a very specific type of incinerator that actually gets hotter than a normal incinerator. I forgot what the temperature is, but it is a ridiculous number to get prions to actually unravel. So prions are kind of scary, in my opinion. Okay. When prions first kind of came onto the scene, nobody knew what it was. Nobody understood what it was. And it took a while before somebody went, wait a minute. And they argued a lot about whether or not this was something that could actually exist. A lot of people thought that it couldn't exist because it didn't have any genetic material. How is it transmissible? It doesn't have any genetic material. Let me tell you, it is transmissible. So yeah, prions are scary. All right. Another form of this is something called Kuru. It was found in Papua New Guinea in a very specific tribe. And in that tribe, one of the things that they did was they would eat the bodies of their relatives. It was a way to keep them with them, whatever their religious belief was. And again, in some way, shape, or form, a prion showed up. And as they died, they ate their body. And the other thing that they did was the parts that they didn't eat, they buried next to their water source. Now, something interesting that came out of that was that one woman had partaken and eaten the brains and everything else, and she didn't get it. So there was a lot of study that went into that woman. Anyway, okay. If you want to see more on that, you can look at the prion video that's on this slide. Okay, so virus-like agents. We've got satellite viruses. They are going to be dependent on other viruses for replication. So they kind of attach to another virus so that they can make copies. Adeno-associated virus originally thought that it could only replicate in cells infected with adenovirus. It can also infect cells that are infected with other viruses, or had the DNA disrupted for some reason. So if you were just to give somebody AAV, it wouldn't do anything. There has to be a secondary virus there for it to actually replicate and make copies. Delta agent. A delta agent is a naked circle of RNA. It's expressed only in the presence of hepatitis B virus, and it can worsen the liver damage effects that hepatitis B virus inflicts. So if we've got delta agent present with hepatitis B, the damage to the liver is even worse. Okay? So, viruses in human health. Viral infections cause billions of infections worldwide each year. The acute infections, acute being short, include things like colds, hepatitis, chickenpox, influenza, herpes, and warts. Geographically predominant infections, you've got things like dengue fever, Rift Valley fever, yellow fever, that you only find in certain places. Viral infections with high mortality rates, rabies, HIV, and Ebola. Ebola is a scary disease that kills people by making them bleed out. HIV, we can slow the progression of that virus, but it's still there. Rabies. Rabies is one of those things that you get bitten by something if it even if it doesn't have rabies, you go through a sequence of shots. It's a really long sequence and it's painful and you don't like it. But it's a way to prevent you from getting rabies. Rabies can be treated if you catch it early enough. If you don't, they can try and make you comfortable and watch you, and you're going to die. Rabies is a fairly insidious disease. It's kind of scary how it works. Okay. Viral infections causing long-term debility. In other words, it causes some type of disability that'll last forever. Polio is one of those. Polio can cause paralysis of limbs. It can cause issues with eyesight. It can cause all kinds of problems. And the thing is, once you have that with polio, you will have it forever. Back when polio was one of those things that was very widespread, you saw kids walking with canes and braces because they had polio, and it never got any better. Theodore Roosevelt was a polio survivor, and he was in a wheelchair, or he used canes to walk around. And even using the canes, he still had to have braces on his legs because, as a previous to that, I should say, because I don't remember if he was a kid or not, he had polio, and it lasted his whole life. Okay, viruses possibly connected to chronic afflictions. This is a really hot area of research right now, by the way. Type 1 diabetes. They think that it's a viral infection that causes the destruction of the beta cells in the pancreas. Now remember, type 1 diabetes is the one that isn't caused because you're heavy set. It'll show up in kids. It'll show up in healthy adults. And the pancreas, for some reason, just stops producing insulin. And they think there is a link to a virus possibly killing those cells, and then you've got diabetes because you can't make your own insulin. Multiple sclerosis. There has been research done that shows that it may be viral, a virus attacking the Schwann cells, the padding around your nerves that keeps them safe and intact. And as those cells die, it causes the nerves to get damaged. And as the nerves get damaged, then it basically causes the symptoms of multiple sclerosis. Alzheimer's has been implicated, obesity has been implicated, specific cancers. So we talked about cervical cancer earlier with HPV. There are links to some diseases that they believe, and research is ongoing. Like I said, it's a hot topic right now. They believe that it was induced by a viral infection. So, with all of these viral infections, how do we treat them? So, things called antibiotics, yeah, they're for bacterial infections. They have nothing to do with viruses. So, if you have a viral infection, hello, a cold, do not take an antibiotic because it's not going to do anything. Colds are caused by rhinovirus. Antibiotics are not for that. Antiviral drugs will generally block the viral replication, it will prevent it from making copies. But they can have really severe side effects on the human body. So you kind of have to weigh the benefit of taking those versus the side effects. People who have HIV are on antiviral drugs. And when HIV was first becoming very prominent and we were trying to design medications to help them and to prevent the virus from replicating, they would get really, really sick. And they were taking, 20, 30 pills a day. So we've gotten better, don't get me wrong, but it is something that you kind of have to weigh the benefits of. How beneficial is it versus how miserable is your life going to be? Interferon is our immune system's natural defense against viral infections, and synthetic versions can be used basically for the same thing. So if I am a cell in the body and I get virally infected, I can do nothing. It's already in my cell, it's already taken over the machinery, it's already making copies. But I can release a chemical, interferon, at the beginning, that basically warns my neighboring cells, hey, there is a cell next to you that has a virus. You need to take precautions. So the cells that get warned actually take steps to make sure that if the virus gets into them, because it will once I explode and virus goes everywhere, they have certain blocks so that that virus can't use their machinery. Okay? Vaccines are also key in protecting us from specific viral diseases, but not all of them. There is no vaccine for HIV. Okay? But there is a vaccine for chickenpox. There's a vaccine for the flu. There's a vaccine for COVID. Vaccines are a very well established way of preemptively preparing your body for exposure to something. So when you get I'll say, flu vaccine, your body creates an army against the flu. So if the flu gets into your body, I don't have to get sick and start an army from the ground up. I already got an army. I get exposed to the flu, my immune system kicks on, and I've got an army already prepared to take out that flu. Okay? So, focus on a disease, shingles. So, shingles is actually a latent disease caused by varicella zoster virus. This is the chickenpox virus. So, you get chickenpox as a kid, you still have it. It's latent in your body. It's not causing chickenpox anymore, but it's there. So, after an individual recovers from chickenpox, the virus goes latent. And it hides actually in nerve cells along your spinal cord, okay, along your vertebra. And as we age, our immune system isn't so great anymore. So our immunity starts to go down. Of course, as we get older, we increase our stress because we become grown-ups with bills and all kinds of other things. There are also some unknown factors that will reactivate that virus. So remember, it's dormant along your vertebra in your back. Viral replication starts and symptoms start to show. Now, the symptoms, because it goes latent in nerve cells, you are in pain and it burns and there's tingling because as it comes out from around your vertebra, usually on your torso somewhere, you get this blistering rash that basically crusts over. There's pain associated with shingles that can be relatively severe because it's affecting your nerve cells. That's where it hibernates and you're starting to damage those. So, yeah, pain is part of that. It can cause nerve damage. If it damages enough of your nerve cells, you're not going to get them back. So, it can cause nerve damage. And because of that damage, it can also cause chronic pain where it just never goes away. Antivirals like acyclovir and the others can shorten the course of a shingles infection. Excuse me. But if you do get shingles, you need to start this within 72 hours of your symptoms beginning. And if you don't know what it is and you don't know that it's shingles, you may not know that you need to start these antivirals. Now, what can we do so that we don't get shingles? There's a vaccine. Zostavax is one of the vaccines to prevent shingles. And there are commercials about it on TV all the time. It's recommended for US adults over 65 years old. This is going to be our most susceptible population to shingles. It can prevent shingles. It can reduce the occurrence of post-herpetic neuralgia. Sorry. So you will see us talk about vaccines quite a bit. But the reason that vaccines are as effective as they are, like I said on the last slide, is that you can build an army without getting sick, and that army stays there so that if, God forbid, the virus comes, there's already an army there to take care of it. It doesn't have time to really get out of the gate because your immune system is on point as far as taking care of it. Okay, so that's the end of chapter 5.